Sir, kindly unmute yourself.
Okay. Now I am audible to all of you.
Yes, sir. Yes, you are
Okay. A very warm good afternoon to all of you. Myself, Dr. Naresh Rangra, working as Associate Professor, ISF College of Pharmacy. Welcome you all in the ISFCP sponsored dialogue series. So today's guest speaker is Dr. Vineet Mehta. He's an alumnus of uh, Government College of Pharmacy, Rohru, and postgraduate from Kurukshetra University, Kurukshetra. And he attained his PhD from JP University, Vaknaghat, with specialization in neuroscience. So his research areas are Alzheimer disease, CNS complications, type 2 diabetes mellitus, chronic stress. He has attained more than 50 publications, including various patents. And his title of the talk is Screening of Herbal Molecules for CNS Complications, a brief overview. So over to you, sir.
Thank you, Naresh. I hope I am audible.
Yes, sir.
Okay, fine. I will share my screen first. My screen is visible, Naresh?
Yes, sir. Yes, sir.
Fine. I hope slides are moving perfectly.
Still not, sir.
You can see my slides, Naresh. Hello.
Okay, sir. Sir, I am sharing.
Naresh, shall I start?
Yes, sir.
Okay, fine. Very good afternoon to all present here and those who have joined us through YouTube as well. First of all, I am Dr. Vineet Mehta. I am currently working as an assistant professor at the Government College of Pharmacy, Rohru, which is in the Shimla district of Himachal, Pradesh. Naresh, one more thing. You can interrupt me in case there is an issue with the slides or audio connection.
Sir, you can zoom in into the full screen.
Okay. Just let me check.
Okay.
Now is it fine?
No, sir, still not visible. Just try.
Just a second. Is it fine now? Naresh, is it fine now?
Yes, sir. Now moving, sir.
Okay, fine. Shall I continue now?
Yes, sir.
Okay. First of all, I would like to thank the entire organizing committee of ISF College of Pharmacy for giving me this opportunity to express my views here. I was listening to the earlier lecture of Dr. Sunita Singh, where Professor Dr. G.D. Gupta mentioned that it is a lecture series of 101 lectures, if I am not wrong. It is a commendable effort from the entire team of ISF College, and I congratulate them. Today, I will be presenting screening of herbal molecule for CNS complication. I will be giving a brief overview. I would not focus on showcasing my research finding, but I would rather focus more on young faculty members, UG students and PG students, and PhD students, of course, to help in designing their research project efficiently in an economic and time-saving manner. I hope there will be many takeaways from my lecture, and I would like to hear the feedback after this session. Uh, During my PhD, I was a hardcore researcher, and I have learned lots of sophisticated techniques, instrumentation, uh, focusing on genomics, proteomics, CNS pharmacology, molecular pharmacology, and everything. Thanks to my PhD supervisors, where the funds were in sufficient amounts, lab facilities were good. But everything changed when I joined this institute. This is a undergraduate institute. The facilities here are of undergraduate level, of course. Being we are a researcher, we don't want to change. When I started here, so I saw okay, there is no facility and what to do. I don't want to get out of my comfort zone. Then I started writing projects, but then COVID hit, and all the fundings are almost dried up. Then I need to shift to some alternates where I can continue my passion for research. And there, at that time, this project design I came up with. So basic problem that I faced, and I'm sure that all the young faculty members and student also face is funding and lab infrastructure. Funding, especially after the post-COVID era, has drastically dried up in most of the institutes. And then there is a time constraint. If you are a UG researcher or a postgraduate researcher doing your master's project, you are having a time of 1 year. During that 1 year, you have to design your project, you have to conduct experiments, you have to generate data, analyze them, and then go for the publications so that you get a good opportunity for higher education, maybe PhD or maybe postdoctorate. So these are the challenges. And other challenges that I came to know from various presentations that I took for master student for Viva Voice presentation, that was the rational and effective hypothesis that was lacking, and that is due to the lack of fund or time constraint, I can say. For example, people are developing nanoformulations. Nanoformulation for what? Of a herbal molecule, herbal plant. And if I ask if this plant is effective as such that can be consumed orally, then why? What is the need of that formulation? Likewise, if the student of pharmacology or pharmacognosy, they have selected a medicine plant based upon literature and then performed a study. That is not exact. This not only will create a problem in your project, but when you go for the publications, the editor, the reviewers will point out these things. What was the background of your project? Why this kind of study was designed? So figured this thing out. I started working on CNS complication, which was my the area of specification. If we talk about CNS complication, the majority of research work in India and everywhere on the CNS is focused on either Alzheimer's, depression, neurodegeneration, learning, memory, dementia, etc. All these are the prime focus of the CNS research. The problem with the CNS complication is that whatever disorder you see, may it be depression, may it be Alzheimer, it is not a single pathway that is involved in the clinical presentation of that disorder. There are multiple pathways. For example, in Alzheimer's disease, there is tau hypothesis, there is amyloid beta, there is then cholinergic hypothesis, along with neurodegeneration, then oxidative stress, neuroinflammation, everything. All these pathways, they come together in harmony and give the clinical presentation of this disease. And what are therapeutics? Currently available therapeutics is, for example, say donepezil, rivastigmine, etc. They are focusing on one pathway at a time. They are focusing on upregulating the cholinergic signaling in the brain. And what about other pathways? They remain unchecked. If those pathways remain unchecked, the Alzheimer or any other CNS complication, that it will continue to grow throughout your lifetime. So probably the best solution for this could be that we need to design hypotheses. Yeah, we need to design some therapeutic strategies that are capable of targeting multiple pathways at once. And for this, herbal medicines, yeah, traditionally used medicine give us better opportunities. This is example of Artica dioica. It is commonly known as bichu booty. It is found in upper Himalayan region, commonly found in Shimla region as well. So if you look in the literature, it has been
Sir,
reported.
pardon to interrupt you, sir. Slides are not moving now.
What's up?
The slides are not moving.
You are stuck on which slide, sir?
This is the third slide, sir. CNS complications.
Okay, now can you see the next slide?
No, sir. So one thing we can do is that can I share your slides on behalf of?
That would be fine, Naresh.
Okay.
Please do it.
Okay, sir. Sir, you were on fourth slide, sir.
Go to next slide, or the plant slide, slide number 4.
Okay, sir. This one, sir.
Slide number 4, Nilesh.
Sorry. Sir, this one. This is the fourth slide.
I cannot see.
Okay, after.
I can see still the second slide. Yeah, this one.
Okay, sir.
Okay, sir. I'm working on this plant, Artigo deva. If you go into literature, you will see lots of pharmacological properties of this plant, which include antioxidant and anti-apoptotic, anti-inflammatory, and lots of other activities. And they can be targeted for Alzheimer, dementia, and everything. It has been reported that this plant is effective against Alzheimer, dementia, Parkinson's, everything. But still, till date, not only one single biomolecule have been isolated from this plant that can be effective against these disorders. Why is it so? The reason for this is that any plant, if you say Artiga deva, any plant, even Brahmi that is used for memory improvement, they are having thousands of molecules. Out of those 1,000 molecules, hundreds of molecules will be bioactive molecules that will be actually responsible for the pharmacological activity of that particular medicine plants. So, when we consume this plant as a whole, it shows a very good effect. That is because the multiple molecules works to multiple pathways of that particular disorder. And when we isolate one single compound, it is not having that much potent effect because it is targeting only one or two pathways at once. So from here, I get an idea of this designing my project. Can you shift to next slide, please? So what we did was we designed a project and divided it into 4 phases. The first phase was network pharmacology, second one molecular docking, then in vitro assay, and then in vivo screening. What network pharmacology is, it gives you the network of your plant. Whatever plant you select, based upon the literature, based upon traditional use, or any random plant that you will select, you can put it through the network pharmacology techniques, and it will develop a network for you against a particular disorder, say Alzheimer's disease. This will give you not only that this plant is effective against that particular disorder, but it will give you the biomolecules. Which biomolecules of this plant are responsible for its activity against a particular disorder, and it will also provide you with the pathway through which pathways these molecules could be effective. Then we move to molecular docking. Whatever results, whatever biomolecule we get from network pharmacology, whatever pathways we get from network pharmacology, we confirm our result through molecule docking. In a molecule docking, as you all know, it is a computational technique where various receptors we can uh, screen with our ligands and see whether it can be effective or not. Then we can narrow down the screening process through in vitro assay, and the final molecules that we get that can be screened through in vivo screening process that we will be talking about later. Coming on to next slide, network pharmacology. As I said, next slide, Naresh.
Yes, sir. It is the next.
As I said, network pharmacology. Network pharmacology, it was a term that was introduced in the year 2007 by Professor Hopkins. He also hypothesized that to treat any particular disorder, we need not to target only just one pathway. We need to target multiple pathways at once. From there, the concept of network pharmacology came into existence. What you need to do the study of network pharmacology, you need just a computer with an internet facility. That's all. Only this thing you need. Then I have listed a few online platforms which you will need to perform network pharmacology, which include your PubMed, PubChem, database, Dish, GNet, CatMapper, and all these. I have also provided the links from you can access these pathways. Now let's see what we get with this network pharmacology approach. In the next slide, as you can see, this is the network image. Move to next slide, please. This is the image of the network that was generated through the network pharmacology. In the middle of this, you can see my plant. It will not be clearly visible. If you zoom into it, when you can see it fine. In the middle, in red color, it is my plant, and the outer circle outer than this plant, it is the bioactive molecule. Out of thousands of bioactive molecules, only 17 are the bioactive molecules that were associated with Alzheimer's. I have prepared this network for specifically for Alzheimer's disease only. And which pathways were involved? The outermost orange circle gives you the pathways that are involved in the that could be targeted for the management of Alzheimer's. So basically, what I mean to say is, Artichola is a plant which is having a 17 bioactive molecules that can be used for the management of Alzheimer's through all these pathways that are highlighted in orange color. Move to next slide, Naresh. This is the next slide, Naresh, slide number 8. This is the protein-protein interaction image where you can see multiple proteins. Those are interacting with each other. These are the proteins which these bioactive molecules of Artichola has targeted to exert the beneficial effects during Alzheimer's disease, or we can say that could be targeted for management of the Alzheimer's disease. Coming on to the next slide. Next slide is the slide where we see the bloom plot of the enrichment analysis. Please show the next slide, Naresh. This is the enrichment analysis plot. The circle depicts the number of targets, the number of genes that are targeted by the bioactive molecule. As you can see, the second last of them is Alzheimer's disease. A total of 20 genes are directly targeted by the 17 biomolecules of Artichola during Alzheimer's. However, if you look into the pathophysiology of Alzheimer's disease, it is the combination of multiple pathways, which include your oxidative stress pathway, inflammatory stress pathway, then is neurodegeneration and everything. If you combine all these pathways along, you get a very large number of targets that was shown in the previous slide in orange color. So from here, you can see your plant is active against which disorder. If you want to work on Alzheimer, you can work on Alzheimer. See here, pathways in cancer, more than 25 genes are being targeted by these biomolecules which are active against cancer. So you can select your disease, your disorder based upon your network pharmacology. If we see what kind of result, what kind of signaling these molecules are targeting, which can be seen in the next slide. This is the network pharmacology image where the various pathways of Alzheimer's disease. These are the pathways of Alzheimer's disease. All these pathways combined together to give the clinical presentation of the disease. The target which are depicted in red color are those targets which are targeted by the biomolecules of Artichola. So the biomolecule will act on these pathway and will halt the progression or prevent the development of the Alzheimer's disease. This can not only be done for Alzheimer's disease, but you can select any disorder and you can work with any of those. For example, in the next slide, I have given the pathways that are for depression. Next slide, please. So this is the pathway of depression. Here also same. What I have to do is, instead of selecting the Alzheimer's disease in my network, I select depression. The software will automatically calculate which biomolecules can be effective against depression and which target they are going to target. So the target depicted in red color are those targets which the biomolecules of Artichola will target for managing depression. So coming on to the next slide, the overall conclusion of the network pharmacology study as of now is that we have screened 17 active phytoconstituents of Artichola, which works on 113 genes that are related to Alzheimer's. So out of those 17, I have just depicted 4 over here, and these are the target genes on which they target, and the disease, of course, is Alzheimer's. You can change it as per your preferences. So now coming on to the next slide. Next slide, in next phase of the project, that was molecular docking. Now, from our network pharmacology, we know that there are 17 bioactive molecules. Next slide, Naresh. There are 17 bioactive molecules, and I also know which targets they are working on. So I will perform a molecular docking study. Molecular docking study is a computational technique where we see the interaction of ligand with the receptors. Naresh, next slide is not visible. Am I audible?
Yes, sir. You are audible.
Your slides
Sir,
are moving. Please move to slide number 13.
slide number 13, sir.
Yes, next slide. So, molecular docking is a technique where you just on the computer on the software you just see the interaction of ligand with your receptors. How to find a ligand? The network pharmacology has already given you 17 ligand of Ortica. Those are the bioactive molecules. And what are the targets? It has given you 117 targets. Out of those, you can select the best suitable targets. What you need for molecular docking is, again, you only need a computer with an internet facilities. The rest, all of these software that I have mentioned over here, Marvin Sketch, Autodoc, Bimol, everything, they are open source and they are free to use softwares. So let's move to the next slide. These are next slide depicts the images of the receptors that we have selected for our study. I have basically selected 5 receptors, but just 3 of them are shown here: acetylcholinesterase, butyrylcholinesterase, tryptophan kinase. Now, if you see, there are lots of software for molecular docking studies. Discovery Studio, Glide, Schrodinger, etc. All of them are paid, highly paid, in lakhs they cost. And then when we talk about Autodoc, it is a free software. You can use it manually by all yourself. The result of Autodoc are equally accepted as by other software. The difference between Autodoc and other softwares is very simple. Those are paid software. You only need to add your ligand, add your receptor, and everything is done by the software itself. However, when we do docking through Autodoc, we need to prepare a ligand manually, we need to prepare a receptor, we need to perform docking manually, and then we need image generation that is also done manually. That's why it is a free software and that is a paid. When we come to the results, that is there is no difference between the results of Autodoc and other software. Even Autodoc is considered to be one of the best software for molecular docking. Now let's see the results. What kind of results we get from molecular docking. As you can see in the next slide. Next slide, Naresh, please. The result of the docking studies are given in form of the energies, kilocalories per mole. The stable the molecule is, stable the receptor ligand complex is, lower the energy will be. As can we have used donepezil as a standard, and we have 3 protein target. One is your tau protein kinase, then we have acetylcholinesterase, then we have butyrylcholinesterase. Let's take example of acetylcholinesterase. As you can see, the docking score of donepezil is minus 11.2. If you see the Herbal molecules, 6 Herbal molecules are having better score than donepezil against this particular receptor. If you see the other receptors, tau protein kinase or butyrylcholinesterase, all the Herbal molecules have shown better result than this standard drug. So what you have to do is that screen all your bioactive molecules that you get from network pharmacology, and you can select number of target, as many targets as you want. You can select 3, you can select 5, you can select 10, 15, depending upon your study, whatever you want. In this imaging you have to do manually. For imaging you have 3 options. Either you can do imaging on Autodoc, either you can do imaging on PLIP server, that is online server, or you can use Pymol software for imaging. Let's see how the images of this docking studies looks like. Please move to the next 2 slides, Naresh. So these are the images, and in the images you can clearly see which amino acid of the receptor are involved in the interaction, what is the bond length, which kind of interaction is it, whether it is hydrogen bond, whether it is electrostatic interaction, whether it is van der Waals interaction. The point here is you only need your docking score, the amino acid which are involved, and plus the bond length. This is and plus the hydrogen bond that are involved, that are most important for your study. So far we have discussed briefly about network pharmacology and molecular docking. The cost of both of these studies is technically zero. You need not to invest anything on these studies. You only need a computer and an internet connection. And what time it takes? Network pharmacology, if you are performing, it will take you hardly 10 to 15 days to complete. If you are doing molecular docking, it will also take you hardly 10 to 15 days complete. Within 10 to 15 days, you will get a good data which suggests that which plant is effective against a particular disorder through which pathway and which biomolecules are involved in it. As you can see in docking study, still we are getting 12 molecules that Sorry for the interruption. I think I lost my connection. Am I audible, Amaresh? Am I
Sir, you
audible?
are audible now.
Okay, fine, fine, fine. So I will continue like this only then. So as of now, as I was saying, the molecular docking and network pharmacology, it just cost you nothing, just your time. That in within 1 month time, you will be having an information about which plant, which bioactive molecule, which pathways for that particular disorder. Now you can focus on your in vitro assays. So as of here, if you are having a cell line facility in your lab, in your institute, you can buy a neuronal cell line which costs you only just 500, and you can go for neuroprotective activities. What you have to do is grow your cell line, add mercury chloride to it. Mercury chloride is a neurotoxic that will reduce the number of viable cells. If your drugs are effective against are having a neuroprotective effect, you will see the increased cell viability. So what about neuronal survival? If you want to check the neuronal effect of your drug on neuronal survival, what you have to do is just simply incubate your cell line with your drugs. If you are getting good number of connections, good number of viable cells, which means that your drugs, your molecules are having a good neuronal survival activity. Likewise, Comet assay is also one simple assay. If you don't want, you are not having cell line facilities in your lab, you can go for Comet assay. It is a assay for genotoxicity. It will tell you about and process like things. For this, what you need is a electrophoresis and a high melting agarose, low melting agarose, and 1 drop of blood. Just perform and commit assay on this, add your drugs, and you will see the result for yourself. For example, as you can see in the image over here, H2O2 was the chemical that was used to induce DNA fragmentation. Olive tail movement gives you the extent of DNA fragmentation. If your molecule is effective, the number of tailing effect is reduced. So you can clearly see from here how your drugs are working. Based upon the result of molecular docking, network pharmacology, and in vitro assays, you can select your best molecule that can be used for the management of Alzheimer's disease. Now coming over to the next slide, the next part of this project. Next part of this project is, of course, now we have screened and Herbal molecule. We have screened and Herbal molecule through this network pharmacology, molecular docking, and in vitro assays. Now we need to study this molecule on animals, how this molecule behave in the living system. For this, of course, if you need to conduct animal studies, you need to get clearance from Animal Ethics Committee. They will simply ask you for rationale. Why have you selected this molecule? Simple. The rationale behind selecting the molecule is simple. You have performed a study. Your study has shown a good result. You can say this molecule is effective in this or this disorder through this pathway. That's why I want to confirm my results through your animal activity. I will not go into the detail of animal activity. You all know animal activity. You will develop a model. You use a model from the literature. Then what you do is test your drug, perform behavioral studies. What after that? After behavioral study, most of the studies are limited up to your behavioral studies, and people go for publication. That is not good for high quality publications. So what we can do is, if you're not having a facility for proteomics or genomic studies, you can just go for two simple techniques. One is your histopathology, and the other one is your Golgi-Cox staining. Histopathology, what you do is sacrifice animals, take out the brain, put it into fixing solution. That is your formaldehyde solution. And you can either make the section if you are having cryotome and microtome. Just prepare a 5 micrometer section and go for hematoxylin eosin staining or chrysal violet staining. If you're not having this facility, doesn't matter. You can outsource this. Every pathology lab charge approximately 500 rupees for histopathology and give you 15 to 20 slides that are more than sufficient for your results. Now let's see the results. What results we get from the histopathology study. Please move on to the next slide, English. These are the results of histopathology. I have focused my study on hippocampus. So these are the three regions of hippocampus: CA1 region, CA2, and CA3 region. This staining is of chrysal violets. Chrysal violet staining is very good staining. It is a lot more better than the that hematoxylin and eosin staining. As you can see, third row, all those dark spots are those neurons that are dead or the neurons that are degenerating. So when you animals with a drug, if your drug is effective, your neuron will appear healthy. As you can see in the control slide, last slide, neurons appear a lot more healthy. What we have to calculate, yeah, and is result. So better approach, please go to the next slide. Better approach than histopathology, because in histopathology you are only seeing whether neuron is degenerating or neuron is healthy. The better approach, in my personal, Golgi box training. With Golgi box training, you can see lots of data. You can get lots of data, and the cost of performing Golgi box training is technically zero. You need nothing to do this. Cost is involved in it. What you need to do is you prepare a Golgi box solution, which is made up of three chemicals: mercury chloride, then you have potassium bromate, and potassium sulfate. You sacrifice animals, take out the brain, and put it into the Golgi box solution. Simple. Incubate it for 37, so for 1 day at 37 degrees Celsius for 24 hours, and then manually you cut the sections. Everyone, I think, must have prepared the TS of leaf root in the pharmacognosy experiment. Same you have to do here. You have to manually cut the sections of the brain, and what you get is the image as I have shown in this slide. As you can see in this image, this number 1, the first image, it is a healthy neuron. See how beautiful it is. It's having a soma in between, number of primary branches, number of secondary branches, and how long the dendrites are going. And if you see the disease control, that is STZ, number 4, you can only see soma and few primary branches. Length of branches has been decreased significantly. This is the severe neurodegeneration that we are seeing in this image. So when we analyze these results as depicted in the graphs, number of primary branches. Number of primary branches, there is no difference. Number of primary branches that are arising from soma. As you can see in healthy neuron as well as in degenerated neuron, there are 3, 4, 5, 6 primary branches. But now come on to the graph number 3. See, number of branches that are reaching 100 micrometer length. The more the length of these dendrite, more the strong, more the is. In disease control, you cannot see this. However, in healthy neuron or in treated neuron, you can clearly see how beautiful these neurons look like. Now, coming over to the slide, zoom it into 100x. This is the image taken on 40x microscope. The next slide, it is image of 100x. Next slide, Naresh. So look, just have a look at this image. This is nothing. The previous image was at 400x. Now this is taken with 10x micro eyepiece and 100x objective lens. You can see very clearly there are dot-like structures on these dendrites. These are called spikes. Spikes or spike, we can say spike density. The greater number of these spikes are present on your dendrite, the more the tendency or more the healthier the neuron is, and more tendency it has to create new connections. Now have a look at the disease control, that is STZ. You cannot see any spikes over there because neurons are degenerating. Neurons are either dead or they are degenerating. They are having no power, no potential to generate new connections. So these animals have performed very poorly in behavior studies as well. The more the number of these grape-like structure or spikes on your dendrites, the more effective your neuron is, the more healthier your neuron is. Now coming over to the next slide, I would like to show you a few images how this Golgi box staining actually looks like under the microscope. See how beautiful this image is. With zero cost, nothing, with a blade, scale, and section. But of course, we need a good quality of a microscope. Coming over to the next slide is again Golgi box staining. Please come over to the next slide, Naresh. So these are see how beautiful neurons can be visualized by using this technique. It costs you nothing. Come over to the next slide. So as of now, I have discussed with you with how cost-effective manner. You use network pharmacology firstly. It costs you nothing. From there, what you get is this plant effective against a particular disorder through which pathway and which biomolecules are involved. Then you move to next. That is your molecule docking. That also costs you nothing. That give you actually which biomolecule are effective against which target. Then you can screen down the narrowing process through in vitro assays. In vitro assay will cost you something with 5,000 to 10,000 maybe at the most. Then animal activity. By following this technique, what we got so far, I was doing regular 5 years I did my regular PhD, and from my 5 year of work, from following the traditional techniques that we are using in lab, I managed to I think 4 or 5 publications in total. Of course, the good quality publication. Now, following this technique that I have just demonstrated you, that cost me nothing, has given me 5 publications in last 2 years. That are also in the good impacted journal, 5.5, 5.3 impact journals, and two are under process. One I am writing. So you can see how beautifully designing your project effectively can work. So I hope I have demonstrated something valuable which you can take home and start planning your research work. Moreover, one thing I would like to, a couple of things I would like to tell all the faculty members, young faculty members, and all the student is that uh, the two things you need to remember. First is read a lot of literature, and secondly, never hesitate to ask for help. If you need to form any kind of study, you just need to identify various lab where people are working on that project. You just approach, don't hesitate to approach the professors over there. There are a lot out there to help you. For example, in my case, the second article that I demonstrated over here was during the COVID era. Everything was fine in that article, but just one reviewer provided me the comments that I have to perform the MD simulation studies. Of course, that facility is not available here, and that facility is also not freely available. So I approached few professors, those were working on this MD simulation, and they open-handedly helped me out. So you need never to hesitate. If anyone would like learn more about the techniques that I have provided here, I am open for it. Please don't hesitate to contact me. So that is all from my end for today. I apologize for the connectivity issue. We have received very heavy rainfall in Himachal over past 10 or 15 days. So there is a lot of connectivity issue. Please, suggestions, any questions, I'm open to it, please.
So, any questions from the audience? Okay. So, sir,
So if there are no,
regarding okay, they can directly contact you through your email ID. I will share it.
okay, you can share my email ID as well as contact details. If anybody want to contact me regarding these technique or anything, any help needed from my end, I'm open to it.
Thank you, sir. Thank you for this mind-opening session on the CNS complications. So, on behalf of IQAC-ISFCP, sir, I would like to extend a really hearty vote of thanks to our worthy expert speaker, Dr. Vineet Mehta, sir, who spared time from his busiest schedule to grace the occasion. And I know it was a hard time as due to rainfall, a lot of internet connection, uh, connectivity issues were there. So today we have an opportunity to hear your thoughts on the topic screening of herbal molecules for CNS complications, and this will surely be going to encourage us in our future research events. So thank you, sir.
Thank you so much, Nagesh. Thank you so much.